Hello to our friends joining us via recording. Today we will be talking about the different levels of structure in our proteins. For a protein to be functional, here's kind of a big picture idea with our proteins. For a protein to be able to do its job, to be functional, it needs to have the right shape. Hey, that relates back to the concept from our very first class together. When I'm talking about a protein being functional, what am I talking about? Which part of our class are we talking about when we say functions? When I say the function of something, I'm talking about its
Physiology.
exactly. Yep, I'm talking about the physiology of my proteins. So if you remember from unit number 1, we said that proteins do all kinds of different things. They make up the shape of a cell, they make chemical reactions happen. All of these things rely on, this physiology relies on the structure or the shape of a protein being right. And remind me again, when we talk about the shape of something or what's there, what was the branch of science that we're doing with that one? What's there, what it looks like. Yeah, then we're talking about anatomy. So when we talk about proteins, their anatomy has to be right for their physiology to be right. They can't do their job if they're in the wrong shape. When we talk about proteins, proteins make up a lot of the space that are inside your cells, something like 20 to 30% of your cells. So proteins are found in a variety of places. I'm going to breeze through a little bit of this because I want to mention here. When we talk about proteins, the monomers or the small pieces that I build them out of are amino acids. So amino acids are a type of chemical. They get their name because of the parts that are inside of them. Let me get my laser pointer for us here. Amino acids have an amine group that's over here on the left side. The amine group has a nitrogen and 2 hydrogens in it. They also have this acid group that we see over here on the side. The acid group has what's called carboxylic acid in it. Now, I'm not going to ask you on quizzes or exams what the chemical formula is for these things, but I'm mentioning to you what they look like because in a moment, we're going to talk about the way that proteins connect or amino acids connect to each other. And it has to do with taking stuff away from the amine group and from the acid group. So we use in protein a very special kind of chemical bond that every time you see these words, you should always be thinking that we're talking about a protein. So something we should all underline, highlight, star in our notes is that proteins are made of with peptide bonds. Peptide bonds. Anytime you hear me talking about a peptide bond, you know right away we're talking about a protein. So the way that I connect all of the amino acids together that build a protein is I use a special kind of chemical bond called the peptide bonds. To build a peptide bond, we do a process called dehydration synthesis. Dehydration synthesis, the chemical process that I use to build a peptide bond. Hey, not a trick question. When you are dehydrated, what's going on with you? If you're dehydrated, what's happening in your body? What's the most basic description of what it
Loss of
means to be
water.
dehydrated? Yeah, so when we get dehydrated, we're losing water, right? It's hot outside, we're sweaty, we're losing water in a variety of different ways. I love that Seth made the connection to our tonic words, right? Those stupid tonic words that we had to learn in unit number 1. Seth mentioned that if we become dehydrated, our tonic words would tell us that inside the cell, we have more water than outside the cell because we've lost all of that water that's outside the cell, which means that inside my cell, looks hypotonic, which is why when I get dehydrated, my cells start to lose water because they're giving water to places that are missing it. So absolutely right. We've lost water when we're dehydrated. Our cells are going to shrink. They're losing water too. When I talk about using the process of dehydration to build a protein, what I mean is I'm going to take away the parts that make up water and build a molecule of water, but also build a chemical bond, also build a way to attach these things together. So the way I build a peptide bond is I go over here to one of the acid groups on one of my amino acids, and I go to the amine group on another, and I say, hey, if you give me an OH and you give me an H, I can put those together to make H2O, to make water, but then I can also connect together the carbon and the nitrogen. So dehydration synthesis is when I build a chemical bond by taking away water. The other thing, we're not going to talk a lot about this in this chapter, but I'll mention this for you because it'll be important later. We can actually, not the highlighter. We can actually do the opposite of this as well. So when we do dehydration synthesis, we pulled out that water to build a chemical bond. You can actually add water back to do a reaction that's called hydrolysis. So I'm going to put that in the chat for us. There's a thing called hydrolysis, which means I'm using water to break a bond. So when we start talking about the way that muscle cells use ATP, they use energy money, they do that process called hydrolysis, where they add back water. So we start with this nice chemical bond over here that we just built. If we put water back into it, we can go back to having a separate amine group and back to having a separate acid group as well. So hydrolysis, we break stuff down using water. But when we're talking about building peptide bonds, we would do what's called dehydration synthesis. We talk about proteins and we think about them as a big long line of the amino acids that are coming together. We're talking about the primary structure of a protein. So the primary structure of a protein is the most basic level of protein structure. Here's an important idea for us to underline, highlight, star somewhere in our notes. We have to get primary structure right for the other levels of structure to be correct as well. So if we mess up primary structure, not always, but most of the time, that can be really scary for the other levels of structure that come on top of it. So primary structure, the basic order that I put the amino acids together in. Notice in my picture, we can see these peptide bonds. So the peptide bonds connect one acid group of one amino acid with the amine group of its neighbor. And then it's also going to form with its own acid group a peptide bond with the amine group of its neighbor. So primary structure, sometimes we call it like beads on a string. It's the line of amino acids that we see. When we put those amino acids in a line, sometimes their amine groups and their acid groups get attracted to each other. The reason they get attracted to each other is because we have dipoles when I form the bond right here between a nitrogen and a hydrogen, or between a carbon and an oxygen. Hey, let's do a little bit of application from what we talked about in unit number 1. If I'm using dipoles to connect these things together, what kind of chemical bond forms when two dipoles are attracted to each other? Do we remember what the name of this bond here in yellow would be? Where I've got a positive dipole up here and a negative dipole down here that are attracted to each other. Yeah, we got several different options. When I talk about dipoles being attracted to each other, that is hydrogen bonds. So hydrogen bonds, I've got the hydrogen up top, the H, and it's attracted to an oxygen down below. This is a hydrogen bond. Now, what gets me to the hydrogen bond is what some of us put in the chat. When we're talking about ending up with a dipole, that means I'm not sharing my electrons equally if I have a dipole. What was the kind of chemical bond where I share electrons, but I don't share them equally? What was that kind of bond called? A sharing bond, but an unequal sharing bond. Exactly. Several of us mentioning in the chat, the way that I get dipoles is I have a polar covalent bond. So between this nitrogen and the hydrogen who's attached to it, the nitrogen gets the electrons more often than the hydrogen does, which means that that hydrogen is going to have a slightly positive charge. Meanwhile, farther down in my amino acid chain, I've got a carbon and an oxygen. My oxygen sees those electrons more often than the carbon does, which gives this a negative dipole. So I use polar covalent bonds to make these dipoles, positive or negative. When the dipoles are attracted to each other, that's what we call a hydrogen bond. So one of the ways that I can make hydrogen bonds between my proteins is this structure that we see right here called an alpha helix. The best way to think about alpha helices is, you know, those birthday present ribbons that spiral around on each other? That's what an alpha helix is. So when my amino acids curl around each other, alpha helix, sometimes they fold kind of flat like an accordion. So you'd see a line of amino acids that bends back and kind of weaves back and forth. It's still hydrogen bonds, just like it was before. It's still the amine group interacting with an acid group over here. It's just that the spacing of these amino acids made it so that they connect to each other in this flat surface as opposed to in a ribbon. So the two different secondary structures that we see in proteins are those alpha helices, the curlicue ribbons, and the beta sheets, or sometimes you'll hear them called, like we see here, beta pleated sheets. We've got an accordion and we've got a curlicue ribbon. That's our secondary structure, alpha helices or beta sheets. Can you remind me again in the chat, what was the primary structure of our proteins? Secondary structure is this stuff. What was that primary structure of proteins? Danae's right. Yep, it's the sequence of the amino acids. It's the order that I put them in. And remember that that order comes together, like Dusty mentioned, from those peptide bonds. So we use peptide bonds to build our primary structure. When we talk about secondary structure, like we see on this slide, we're using hydrogen bonds to build that. So we're attracting dipoles to each other. When we look at this protein that we see on the screen here, notice that we've got areas that have alpha helices and beta sheets. The two types of structure are present in different colors. Can you tell me which color would represent the alpha helices in this protein? Where are the places that I see the alpha helices? What color is that? Yeah, we're like, I don't know what to call that color, right? I have a colorblind, I have two colorblind brothers actually. So all the love for anyone who happens to be colorblind. Yeah, the orange, yellow color, whatever we want to call this that we see here, those are my alpha helices, my little curlicues. And then when we look at the blue, notice that the blue has things that are flat. Things that are flat are those beta pleated sheets. When you look at this protein, notice how there's some areas that are just this gray line that we see here. When we think about secondary structure on a protein, not all parts of that protein might have secondary structure. Some parts might just have a straight line. So it's only when the amino acid chains interact in a particular way that we generate the alpha helices or the beta sheets. Some places might just have them strung up in a line. When we look at this protein, though, it's not only that I have the beta sheets in blue and the alpha helices here in this orange gold color. I took all of those things and I folded them together into a 3D shape. So what we're looking at with this protein is something we're going to explore here called tertiary structure. So when I talk about a protein folding into this shape, this happens because of one other part of the amino acid that we were ignoring earlier. Yeah, so Danae asked a clarifying question. If beta sheets are always flat, that's correct. The way that those amino acids interact with each other is in a flat surface. Alpha helices are always the curly Q ribbons. Any other clarifying questions? I've been talking too long, right? Any other clarifying questions? Or give me a thumbs up if you're tracking with me so far. I got one thumbs up. I got one friend tracking with me. Good. There we go. Okay. So we're talking about a protein. Oh, Seth is very enthusiastic. I got a great thumbs up from Seth. So that means we're good. So far, we've talked about primary structure. It's a line of amino acids in order. Secondary structure, like we're seeing here where we've got the alpha helices and those beta sheets. When I take all of those things and I take them from just being, think of them flat on the desk in front of you, when I take them from being flat and I curl them up into this 3D shape, like we see here on the slide, that's the thing called tertiary structure. So yeah, Hugh asked a clarifying question. Tertiary structure is a result of primary and secondary structure, or kind of a combination. Yes, that's accurate. The tertiary structure, this 3D shape that we see, we can't get that 3D shape unless we get our secondary structure right. And we usually can't get the secondary structure right if we don't get the primary structure right. So let's talk about how we go from the flat ribbons and the curly Qs to something that's 3D. That has to do with a part of the amino acid that we didn't talk about yet. And that part is this part up here. So this part up here is the functional group of an amino acid. Hey, this is not a biochemistry class, so I'm not going to ask you to memorize the structure of all these different functional groups. When we talk about the functional groups, though, they're very important because, to use technical anatomy speak, this is what gives an amino acid its biochemical properties, which is a fancy word for saying, how do you feel about water? What kind of chemical bonds can you make? Those things are biochemical properties. I like to think about that as the personality type of an amino acid. So I can't remember if I've mentioned this to you all or not. Surprisingly, maybe to you, Dr. Ellis is actually an introvert. I really like to be alone. I'm not really like a people person, even though I'm a teacher. It's a weird fit. But if we're an amino acid, I don't have personality type of an introvert or an extrovert. The personality type that I have is polar or nonpolar or a positive charge or a negative charge. So the personality types that we have when we talk about amino acids are not the same as human personality types, but we can use them kind of in the same way. Because if I know the personality type, or technically the biochemical group of an amino acid, that can tell me a lot about it. So again, I've given you the structures of the different amino acids. I promise, I will not ask you to ever draw them or to identify them from a list. But I wanted to make sure to show them to you because the structure of these chains is different. The chemistry of these chains is different, which is why they're the different personality types. So I've got a group up here at the top of 9 different amino acids that are non-polar amino acids. Remember when we built that thing on the board, that list on the board that showed all the things that non-polar meant? What were some of the things that we said that non-polar means? If I tell you these are non-polar, what can you predict based on that? Yeah, so several of us are accurately remembering that if I'm non-polar, I'm hydrophobic. I do not like water. My non-polar amino acids do not like water. My non-polar things also have no charge. Yeah, because as someone else mentioned, we've got the electronegativities that are similar to each other. We're sharing those electrons equally. So when I have an amino acid that's non-polar, the kind of chemical bonds I would find inside of it are non-polar covalent bonds. Those are my equal sharing bonds, which means I don't play nice with water. But some of my amino acids are actually polar. When we talk about things being polar, now it's all the opposite, right, of what we said for non-polar. So if I'm polar, I do or don't play nice with water. If I'm polar, do I play nice with water? Yeah, if I'm a polar molecule, I do play nice with water. Remember that polar means I have dipoles. Water has dipoles. Water has partial positives and partial negatives. Since water has dipoles, if I have dipoles as well, I'm going to play nice with water. Remember that things get polar when they're not sharing the electrons equally. So notice how down here I've got an oxygen and a hydrogen. Oxygen loves those electrons more than hydrogen does. Oxygen is going to be slightly negative, hydrogen slightly positive. Same deal over here. When we get over here, we've got a sulfur and a hydrogen. Again, that sulfur wants the electrons more than the hydrogen does. So all of these polar ones play nice with water. We have some that we say are charged, meaning they have a positive or a negative charge. Why we care about these positive and negative charged amino acids is because the kind of chemical bond that we can form between a fully positive and fully negative thing. What was the name of the kind of chemical bond where you're negative, I'm positive, we're attracted to each other? What was the kind of bond that was just based on opposites attract? No electron sharing, just electron stealing. Exactly. Yeah, several of us chiming in. Ionic bonds. So when we talk about positive and negative, negatively charged amino acids, we're talking about the kind of amino acids that can form ionic bonds. So we care about these guys because we're going to want to have a negative close to a positive. They're attracted to each other. They love to be close to each other in the protein structure. So my personality types of my amino acids are what come together to make the 3D shape of the amino acid. So let's look at the various kind of bonds or attractions that lead us to fold into a 3D shape. So let's start over here on the left. We're going to start with hydrogen bonds. Now remember when we talked about hydrogen bonds just a bit ago, that these form between dipoles. Hydrogen bonds also are not the strongest bonds, which is why I put them in quotes for us. They're not sharing electrons. It's the attraction of one positive dipole to another negative dipole. What's making this different is the fact that my amino acid over here is who has the positive dipole, and many, many, many amino acids later is the one that has the negative dipole. When we before were talking about secondary structure, it was amino acids that were pretty close together. When we start talking about tertiary structure, we're interacting things that were really far apart. So the hydrogen bond between an amino acid up here and the amino acid down here, that's going to fold this whole protein from being just one flat long chain to kind of folding up on itself. So hydrogen bonds, one way we can build tertiary structure. Another way we can build tertiary structure is what we see over here, called hydrophobic attractions. Hydrophobic attractions are not an actual kind of bond or attraction in that way. Hydrophobic attractions are essentially, we all hate the same thing, so we're going to squeeze together. When I talk about hydrophobic attractions, what do we all hate? What do these amino acids, what are they afraid of? Let's put it in our terms from before. If I'm hydrophobic, yeah, exactly. I'm afraid of water. So if I have one amino acid up here that doesn't like water, and a little ways down my chain, there's another amino acid that has the functional group that doesn't like water. When I squeeze these things together, they're like, hey, you don't like water. I don't like water. Let's stay close together. That will help us all to be happy because we can push all the water away. I kind of like to make the analogy with hydrophobic attractions to sports teams. So I'm going to be fully honest. I did not watch the Super Bowl this year. I didn't care at all about the Super Bowl this year. But I do have a football team that I love to root for on occasion. And that team is the Denver Broncos. That is what I grew up with. So I do love me some Denver Broncos. They've been really bad lately. I'm hoping for a miracle eventually. I root for the Denver Broncos. They're the team that I like to see winning. I suspect that there are several Cowboys fans in the house. Any Cowboys fans in the house? We're in Texas, so I presume so. Yeah, Laura likes the Broncos too. Love it. We all don't like the Cowboys? Okay, one Cowboys fan. Okay, there we go. Couple more Cowboys fans. So on a normal week, some of us are rooting for the Cowboys. Some of us are rooting for the Broncos. As you go up and up and up in the hierarchy of games, we go to the playoffs and, you know, the wild card and all that kind of stuff. As your team drops out, you start to develop like anger against a team that beat your team. So maybe you usually root for the Broncos, but the team that beat the Broncos was the Raiders. We love to hate on the Raiders as a Broncos fan. So it doesn't matter. So if the Raiders make it all the way to the Super Bowl, I don't care who they're playing. I'm rooting against them. I don't like the Raiders. I'm going to root for whoever else is the team that's playing the Raiders. It's the same way here with the hydrophobic attractions. We may not have anything else in common, except we all don't like water. Because of that, we're going to squeeze together and we're going to make a part of my amino acid where there's no water. So think about a group of people coming behind some sports team that maybe they didn't root for all season, but they root for it at the very end because they want to root against somebody else. Yeah, sorry, Alejandro. It's just a rivalry that goes deep, right? Alejandro likes Raiders. So nothing but love for you, but go Broncos. All right, so we have talked about hydrogen bonds between the dipoles on these two amino acids. We've talked about hydrophobic attractions between two amino acids that don't like water, so they squeeze together to keep the water out. Another kind of bond that we can form to build proteins are called the nonpolar covalent bonds, or in proteins in particular, we see a special kind of them called disulfide bonds. So if two of my amino acids, one over here and one way over here, both have a sulfur in them, those sulfurs can interact with each other to make a disulfide bond. Based on the fact that a disulfide bond is a non-polar covalent bond, do you think that disulfide bonds are strong or weak? Would you predict that a disulfide bond is strong or weak? What do we think? Yeah, several of us are correctly chiming in to the chat. This is a non-polar covalent bond. This is the strongest kind of bond that we can build. Not only am I sharing, which is what covalent means, but I'm sharing equally, which is what non-polar means. Non-polar covalent bonds, which is the kind of bond that a disulfide bond is very strong. So when I connect these amino acids together to one another, they're going to stay stuck. They're going to really help me keep this 3D shape going. My hydrogen bonds are going to be a little bit weaker. They will help a protein to generally stay folded, but we got to watch out for those. Hydrophobic attraction, same deal. If they find another amino acid that's hydrophobic, they might move a little bit, but there's no moving with the disulfide bonds. One other kind of bond that we can build between that last personality type that we talked about, and that's the ionic bonds. So if I have an amino acid that has a fully positive charge or a fully negative charge, I can make an ionic bond between those things. So your total positive attracts my total negative. We're going to stick together at least until we're exposed to water. Remember from unit number 1, if I expose this to water, it may not stay together anymore. There's something else that has a positive or negative. I may end up going that direction. So all of these different kinds of interactions, whether it's the hydrogen bonds, whether it's the hydrophobic attractions, these disulfide bonds or ionic bonds, all of these things that make my protein fold into its 3D shape, this is that stuff that we call the tertiary structure. So the 3D shape of a protein comes from those functional groups or the personality types of my amino acids interacting with each other. I want to pause for a moment and give you a chance to ask any questions, or if you have any clarifying statements you want to make. Let me pause for a moment as we wrap up tertiary structure. Yeah, so Danae's asking for a review really fast of the disulfide bonds. The two things we should know about these is when I have, the first thing is they form between these sulfurs. That's what S is. These two sulfurs are on amino acids that are far from each other. Because they're forming a bond with each other, they both want the electrons the same amount, which means they're going to share them equally. So we're going to have a non-polar covalent bond. And those are the strongest kind of chemical bonds that we can make, non-polar covalents. Shoot me a thumbs up if we're good, or some other emoji. We haven't done like an emoji fest yet in our class today. I like it. Shelby's feeling a little moody. I feel that. I'm going to get my favorite. Lum sent me a fun GIF. I'm feeling that too, Lum. I'm about ready to fall asleep myself. All right. Let's talk about, I'll mention, I'm not going to take class time for this, but you have this activity in your notes outline where it asks you to compare all the stuff we've talked about so far. So the different parts of the amino acid that are involved in forming these different structures, as well as the specific kinds of chemical bonds that are used to form each. So as you're processing through what we've talked about in class so far, come back to this stop and think it through activity. Make sure we can identify which of these levels of structure are built using peptide bonds, which are built using ionic bonds or covalent bonds. So use your notes to help you fill in this table. Wink, wink, nudge, nudge. This is an excellent thing to help you with studying. So if we can remember which kinds of bonds and which parts of an amino acid contribute to each of these levels of structure, that will be very helpful for you on the homework assignment and on future quizzes. In addition to giving our proteins their 3D shape, though, the other thing that my functional groups do is they give me an idea of where I would find my proteins when they're in the plasma membrane. So several of the proteins that we have in our cells have either part of themselves or all of themselves embedded in the plasma membrane. When we think about the kinds of amino acids that can be on the inside or the outside of the membrane, that's different from the kinds of amino acids that can be in the middle of the membrane. And it all comes down to that chemistry stuff that we talked about last unit with the plasma membrane. When we're talking about the part of our membrane, these little tails that we see inside here, were my tails polar or non-polar? What was the chemistry of my tails? The middle part of the membrane. Exactly. Several of us are saying this is the non-polar part of the membrane. Totally correct. Excuse me. If I'm non-polar, I only like other things that are non-polar, that don't have a charge. So if your protein, so this one right here is what we call a transmembrane protein. I'll type that in the chat for us. Transmembrane. That means it goes all the way from the inside to the outside. If I'm a transmembrane protein, some of my amino acids are touching and are right there with those tails. If I'm going to be right there with the tails, I have to match the tails. So on my images here, all of these parts that show the protein in blue, these are amino acids that are non-polar. They have to be able to be touching those tails. The tails don't play nice with water, and they don't play nice with any of my amino acids that do. So I can only put non-polar amino acids inside the membrane. When I am outside of the membrane, on the outside of the cell, or on the inside of the cell, remember that those were places in the cell or outside where we had water. So when we're thinking about our purple colored proteins or magenta, whatever color they are, these are proteins that are surrounded by water. These are proteins that have to play nice with water. So some of the words we could use for these ones, some of the personality types for these ones are going to be polar, or it could be my positively charged or my negatively charged. The big idea being, if you're outside or inside the membrane, you have to play nice with water because there's water in either of those places. How do we feel about using the functional groups, using the personality types of amino acids to predict where they'd be found? Thumbs up, thumbs down, nervous face. What do we think about that? Good. Okay, getting several thumbs up. I will mention for you on the homework assignment and on the exam, I'm going to give you the names of some amino acids, and I'm going to tell you their personality type. So I'll tell you this one is nonpolar, this one's polar, this one's positive or negative. I'm going to give you that information on the exam. You will need to be able to predict who would I find inside here, or who would I find out here or in here. So make sure we can use those personality types to predict where things go. Yeah, so Danae mentioned still a little bit confused. Yeah, I think the more that you look at it, focus on the chemistry. So there's two parts of chemistry. There's the chemistry of the tails and the heads, and then there's the chemistry of our amino acids. If you can remember what's going on with the membrane, that will help you to predict what would go on with the proteins as well. So definitely give it some time to sink in. It's totally fine if it's still confusing at this point. Chemistry stuff, gotta love it, right? Or gotta love to hate it, as many of us would say. There is one other level of protein structure. And I should mention with this level of protein structure that not all of the proteins in your body have this level of structure. So this level of structure is called quaternary structure. For you to be able to have quaternary structure, there has to be more than one amino acid chain that is interacting with each other. So we're looking over here at a protein we've talked about before called hemoglobin. Hemoglobin is my oxygen carrying protein that I see inside my cells. The hemoglobin protein is actually made out of 4 pieces that come together to make the big protein. So let me explain to you how we went from this picture to this one over here. In the hemoglobin protein, there's 2 proteins that are called alpha subunits, and there are 2 proteins that are called beta subunits. Now, don't confuse those with the alpha helices and beta sheets. They're different. It's just the name that I call for this big protein right here. So I'd call this a beta protein. I'd call this an alpha protein. When I'm building hemoglobin, I need 2 alphas and 2 betas. To get those 2 alphas and 2 betas to stick together, I'm going to use the same kind of chemical bonds that we talked about before. So I'm going to use hydrogen bonds and disulfide bonds. I'm going to use hydrophobic attractions. What this picture shows you, this funky one here on the left, scientists went through and they looked at the sequence of all the amino acids that are inside the hemoglobin proteins. And they looked at which particular ones on this protein chain are interacting with these protein chains. So they went through and they said, okay, amino acid number 141, this amino acid is interacting with these 2 in the chain over here. Or this amino acid number 94 in this beta chain, that's interacting with amino acids up above and some that are across. What this picture shows you is it takes a whole lot of different kinds of chemical bonds to hold together an amino acid chain, or excuse me, a big protein. So I've got 4 amino acid chains that make up this protein. I've got to glue them together using all of these different types of chemical bonds. So if I have a protein that's just one amino acid chain that's folded into its 3D shape, I have tertiary structure. But if I take this protein and I add it with one here and here and here, if I now have 4 different amino acid chains that come together to make a big protein like hemoglobin, now I have quaternary structure. So that final level of structure, quaternary structure, if I have more than one amino acid chain. Now, we can have some problems, like we mentioned at the very beginning of this class, we can have some problems if our proteins don't fold correctly or if their structure's not quite right. So for us to be able to have a functional protein for the correct physiology, I have to have the right shape, the right anatomy. If a protein loses its shape permanently, you know, we talked about gated proteins where they change their shape. Change is totally fine. We can go back. If we permanently lose our shape, meaning there's no way I can go back to what I'm supposed to be, I become denatured. And denatured is an underline highlight star word. That's a really important word. Denatured means that I have lost my shape forever. I can't go back to my correct shape. Hey, not a trick question. If I can't go back to my correct shape ever, do you think I can go back to my correct job ever? Would a denatured protein be able to do its job? Exactly. We're all saying no. It definitely would not. Denatured proteins cannot do their job. That's why they're so harmful to us. There are many different ways that you can denature a protein. One of the ways that you're most familiar with is by adding excessive heat. So over here in my picture, we're seeing 2 eggs that are in a pan. We're frying these eggs. When you fry eggs, you take the proteins that are here in the whites of the eggs that are really clear. When I put enough heat on these proteins here in the egg, it makes them turn white. Like they change so much that their shape is so completely changed that now they don't let light through. Now they are white. You can't unfry an egg. That egg is always going to be fried. So when I use excessive heat to denature a protein, it's the wrong shape forever. I can also use things like chemicals. So for example, if I expose a protein to something that is a really high acid, like stomach acid, for example, that's going to completely wreck its structure. Or if it's really basic, like ammonia or bleach, stuff in cleaning supplies, that can denature a protein. Like we mentioned, heat or even extreme cold can mess with proteins. So the big idea is we want to make sure our proteins don't permanently lose their shape. If they do, we can't fix them. They're stuck. But here's what's scary about denatured proteins in the body. Sometimes one protein that's denatured has the ability to go and denature other proteins in your body. So let's look at how this works. In your body, you have a couple of proteins they're called prion proteins. So these proteins over here on the left is their normal shape. Hey, not a trick question. When I look at this normal shaped protein over here, what's the name of the secondary structure that I see here? What did I call these things that look like curly Q ribbons again? What were those called? Excuse me. Exactly. Yep. My curly Q ribbons were called the alpha helices. So normally this protein that I see right here has curly Q ribbon inside of it. It has alpha helices. When this protein becomes denatured, when it permanently loses its 3D shape, here's what it looks like. What is the name of the structure I see now? I don't still have those alpha helices. Now I've got a whole bunch of what? What were these ones called? Yeah, these flat ones that we're seeing here, now I've got a whole bunch of beta sheets. I've got my normal protein in the correct shape and my denatured version of the protein. They have different secondary structures, which means they have different tertiary structures. So I have different shapes at all levels, starting at the secondary structure. Here's what's scary. We got my normal protein. I made my normal protein from my mRNA. That's our topic next week, is how to build proteins. So I make my normal protein. It's chilling out, doing its protein things. And then my prion protein, my misfolded protein, comes over and says, hey, you would look way better if you had beta sheets. And this protein's like, yeah, you seem pretty cool. Sure, I'll get beta sheets. It goes from being folded in alpha helices to being folded in beta sheets. Except now that it has beta sheets, it can't do its normal job anymore. And even worse, now it can go around and it can go find somebody else who's folded correctly and say, hey, you would look way better with beta sheets. Okay, I'll get beta sheets too. This process goes on and on and on until we have so many of these prion proteins that are misfolded inside our cells that it leads to the death of the cells. So you're going to do some reading in your application activity about a prion disease called Creutzfeldt-Jakob's disease. This is a fatal neurological disorder in which a patient gets a whole bunch of those prion proteins that build up in their neurons. This disease is very similar to mad cow disease, which the technical name is bovine spongiform encephalopathy. The emphasis with that is spongiform. That means that there's holes. So you'll see in some of those resources I provided for you what the brain looks like in a patient who has this disorder. There are places where neurons used to be that are now empty. Because the neurons have died, they were too full of those prion proteins. So before we transition into group work, the big idea with protein folding and disease, you have to be the right shape to do your job. If you're not the right shape, if you've become denatured, you might start denaturing other proteins as well. And if I get too many of these non-functional junk proteins inside my cells, that can lead my cells to die. And that's the problem in Creutzfeldt-Jakob's disease, CJD. That's also an interesting connection to make here too. If you've ever heard about how Alzheimer's is a problem with beta amyloid plaques, it's a buildup of proteins that have too many beta sheets. So some of these neurological disorders that cause death of the neurons, some of them are based on proteins that are folded incorrectly, leading these proteins to build up. That's all I have to share with you. I'm going to transition us into group work. But while we're getting ready for that, are there any questions that you have that you'd like to pose in the chat? I am getting our group work as we speak. Well, we're all very quiet, which means I need an emoji. How do we feel about this stuff? We good on this stuff? I liked your reaction, Hugh. Yeah, Amush mentioned that it is super scary. This disorder that you're going to read about, Creutzfeldt-Jakob's disease, is no joke. It doesn't happen very often in the United States, but I did have, interestingly, one semester a student told me she worked at a hospital and they actually had a patient with it. So small world. She had an idea what was going on because we talked about it in our anatomy class. Okay, I have put in the chat a link to our group activity. If everyone wants to go ahead and click on that, I am going to split us up into groups here in just a moment. Remember, only one person in your group needs to complete the form for your group. Once you have completed that group activity, you were done with our lecture class for the week. Remember that next week, lecture and lab are both going to be online. So I will post a link for you on Sunday to both of those times. I'm trying to think if there's anything else to wrap up. There is an assignment for the Creutzfeldt-Jakob's disease as well as the protein folding. So please make sure that you're completing that application activity as well as you're working on the protein stuff. Almost done assigning. I need the dumpster fire Elmo. Perfect. Okay, got everyone assigned. All right, well, I'm going to end the recording. I'm going to open up my groups. Once your group members are there, go ahead and come onto those mics or use that chat. Let's get that activity done.